Welcome, flat earthers. Another happy day of truth, and in particular, I hope to touch on something that's very near and dear to my heart, which is the vegetation experiments which I have provided my preliminary rough sketches. Exactly what happened to the exposed species that I had in the greenhouse. Thankfully, it was vacated long enough where I did have a full week to work with it. But today, I'm going to touch on something within the first post of my experiment. I mentioned the C4 photosynthetic pathways in plants, and it should be noted that these differences, while the obvious differences are quite, large in magnitude, we have very, very, very tiny. It almost looks irrelevant discrepancies between scientists when they go into actually the requirements for a plant to fully go through its photosynthetic pathway. On the C4, it was suggested that there's a difference in their ability to absorb iron from the root growth, and what I'm here to do is elaborate on a recent research product by Henderson Cornwell Smith on the C4 plants. And what he did is he looked at the pasture plants grown in the sand cultures, and he found out that the thermogenic mycotoxins produced by the penicillium proved to be the vital factor in isolated testing. Of course, credit to White for the penicillium species discovery, but it was the thermogenic mycotoxins produced that made the C4 species ready chloric. The chlorosis cannot be attributed to the mycotoxins, but it was only alleviated when the plants were sprayed within 1% of the microgram of the ferric citrate Fe, dominating the discussions within his abstract and introduction. But he does make a downwards trend towards the different experimentation has been used to isolate such molecules and elements, and Hewitt was mentioned in his sand community, in which he isolated the very, very similar elements. And while there are many reports of the iron chlorosis when grown in sand and water, some of them brooking, matching, wise, even in the 1970s there was some of the same nature, and they really looked at the differences in plants that well, statistically significant. But up until this point, there has been no systematic way to individually say that there was a great importance of difference. And in the first experiment within his research paper that I am going to elaborate on, he uses a hydrochloric acid diluting in the sand dehydrated. That's the HCl parentheses AQ, and in parentheses, don't want you guys to somehow think it's a gas or something crazy of that sort. And when he looks at the ammonia, the nitrogen, nitrate, phosphate, sulfur, chloride, etc., etc., he had a 20 milliliter solution displaced from around each of the roots. In preliminary experiments showed that up to 50 milliliters of the solution could be displaced with the deionized water. And when we look at this statistically, because I don't really care that what he found was what he expected, or he found something out of the ordinary that you can't quantify. And when we look at the statistical analysis, we look at the group of each species presented in his tables, and when he has the relationship between the dry matter yield, and we're going to have that as the capitalized Y, lowercase d, and the iron concentration in the nutrient solution, which we will call X, and between iron concentration in the leaf, big Y, big L, and the iron concentration in the nutrient solution X, approximated by the inverse polynomial, YD or YL. Let's stick with the capital Y, capital L, equals X over the whole, in parentheses, the 1 plus bx plus cx squared, end of parentheses, with the parameters a, b, and c. Now this has extraordinary advantages over ordinary quadratic polynomials, and that it is non-negative, bounded, and no built-in symmetry about the optimum, and has an asymptote when C equals 0. And this relationship between the ion concentrations and the roots and concentrations of the ion in the nutrient solution was described by a straight line relationship on a log transformed data. But then you look at the effect of the nitrogen source on the plant species and on pH of the root size. Now, plants grown on the ammonium nitrate-based nutrient solution reduce the pH from 6 to between 4.1 and 5.2, while those grown in a nitrate-only based solution, and those small differences among the plant species. It's really difficult to put your finger on which is the most significant, but I will try and run through what I think is quite important for people to realize, is that by contrast there were C4 plants grown on the ammonium nitrate solution of the pH of the root zone was reduced slightly to a lower range of between 4.1 and 4.4 compared to the 4.4 and 5.2 for the C3 species, which is more common that you would see in the classroom, that a basic biologist would explain to the students. This is the very drawing cut, basic, very, very basic way that photosynthesis occurs in the species, and increasing the ion concentration in the nutrient solution had no significant effect on the pH in the root zone, nor did it interact with the nitrogen source of plant species. So that is kind of a contradiction of terms, if you think. So then you might think to yourself that the embryonic tissue might actually have a role there. And with that, I pose you a simple question: if that is the truth, then why not replicate my own experiment more, which shows the calincomas? Those specific species I chose was very, very, it was not by coincidence by any means. And I hope the rest of you will research more on CAM plants, because that is the next topic I wish to speak about. I really should be cutting it short now. I don't want to keep you too late. I'll make another video soon about CAM plants. You know, pay attention to those pineapples, whatnot. Soger's an idiot, but just keep your eyes on what science is coming out with. Review everything as much as you can. Do your own experimentation, and you will find quite fascinating results. And that science itself, the globalists have a dogma, but they haven't made the best efforts to stop the results of one, so to say, branch or organization from contradicting, the other. And that worries me, because that says a couple of things. One, that the people are not paying attention to science and the publications coming out by respective organizations that really should. The most important thing for a society is to question, especially those that are in the power, and coming out with the laws and regulations within the land. And I think if more people, I dare say, I might get yelled at for saying this, but if you do not convert to believing in a flat earth, at least question what is around you. Question everything, even as basic as the earth's shape. If you question the earth's shape and basic principles taught in first and third grade classrooms, you are in for quite a surprise. And the citizen that questions is the citizen that contributes the most beneficial improvements to a society. So thank you very much, and in veritate victoria.